So maybe you've got this worksheet, or even worse, you're expecting an entire exam page full of macromolecules to identify. Let's work it out. Hi biology friends, it's Sarah, your other biology professor, and let's talk about how to identify the macromolecule structures. Be sure you stick around to the end for a quiz featuring some of the most common macromolecule mix-ups that I've seen from my own students. But for now, let's get into it. How can you tell if a molecule is a carbohydrate? In a carbohydrate, you're going to see the elements carbon, hydrogen, oxygen, and that's usually it. You're going to see these elements in a 1 carbon, 2 hydrogen, 1 oxygen ratio, or something very, very close to that. Okay, so what about the actual structures themselves? Well, the monomers, building blocks of carbohydrates, are monosaccharides. You may see them in either a linear or a ring structure. Both of these are glucose. Notice that you've got 6 carbons, 12 hydrogens, and 6 oxygens. That's our 1:2 to 1 ratio. Monosaccharides stay pretty small, about 3 to 7 carbons, and most of the ones you'll run into have about 5 or 6 carbons, so pentose or hexose sugars. What about when we double it to make a disaccharide? This one's lactose. Still carbon, hydrogen, and oxygen. Now, of course, when we're building polymers, we're going to lose some water due to that dehydration synthesis reaction. So that means in the polymers, you're not going to see that exact 1:2 to 1 ratio, but again, still really, really close. And even longer polymers, polysaccharides, will keep that vibe. The most important biological polysaccharides that you'll run into are all polymers of glucose. Things like starch, cellulose, and glycogen. Again, notice the composition, carbon, hydrogen, and oxygen, and how we're just combining those monosaccharides. A lot of these polysaccharides will be straight chains, but for some, like glycogen and amylopectin, you'll see some branching. This is a very specific example that will not apply to most of you, but if you have been told that you need to identify specific macromolecules by name, and one of those macromolecules is called chitin, a polysaccharide that you find in crustacean shells and fungal cell walls. Chitin looks like cellulose, but with nitrogens, so it's a little bit of an exception to our carbon, hydrogen, oxygen only rule. Let's move on to the lipids. Lipids are a big bunch of weirdos that all have hydrophobic areas. You can identify these by lots and lots of bonding between carbons and hydrogens. Lipids, you're going to again see carbon, hydrogen, and oxygen, but much less oxygen than with the carbs. There's not really a monomer with the lipids, but many of them build off of fatty acids. Now, fatty acids come in 2 flavors, saturated and unsaturated. Saturated fatty acids are going to be a nice straight chain, no double bonds between carbons, whereas unsaturated fatty acids will contain double bonds between the carbons, introducing kinks to the chain. So only 1 double bond, monounsaturated, or multiple double bonds, polyunsaturated fatty acid. 3 fatty acids plus a glycerol gives you a triglyceride, a fat or oil. These guys are kind of shaped like the letter E. So tri means 3 and it's shaped like an E, it's a triglyceride. But 2 fatty acids and a glycerol plus a phosphate group gives us a phospholipid, a key component of the cell membrane. And with that phosphate group, that's the only exception to our CHO components so far. And finally, steroids. Now, you can recognize steroids due to the 4 fused rings. With polysaccharides, we were also attaching ring to ring, but with a bond in between. Whereas with the steroids, these rings are solidly smushed together. Oh, and you'll also notice that due to the hydrophobic nature of the steroids, you're going to see a lot less oxygen than we did with those carbohydrates. Proteins are going to introduce a new element, nitrogen. The monomers of proteins are amino acids. Amino acids are made up of a central or alpha carbon bound to an amino group, there's our nitrogen, and a carboxyl group. And we call this the backbone of the amino acid. They all have this structure. But amino acids also have an additional R group or side chain. The R group varies by amino acid type. It's what gives the amino acid its particular properties. Now, with 20 different standard amino acids, that means that when we combine amino acids to make a dipeptide, two amino acids, joined by a peptide bond, or an oligopeptide, a few amino acids joined together, or a polypeptide, many amino acids joined together, you're going to see a lot more diversity of structures than you did with the carbohydrates, which kind of just look like near identical subunits stuck together. In the polymers, you can look for the N-terminus, the amino terminus, on one end, and the carboxyl or C-terminus on the other end to confirm that you've got the right macromolecule. The nucleic acids will also have C, H, O, and N, but we are adding P here, phosphorus. The monomer of nucleic acids is the nucleotide, consisting of 3 pieces. So you're going to have a phosphate group, just like in the phospholipids, a monosaccharide, that's right, monomer and a monomer here, as well as a nitrogenous base. DNA and RNA nucleotides utilize different 5-carbon sugars, deoxyribose for DNA, ribose for RNA. And those nitrogenous bases can come in 2 different varieties. Could be a single ring, or pyrimidine, or a double ring structure, a purine. Now notice, double ring, just 2, not 4 like with the steroids. 5 different bases, A, C, G, T, and U, give DNA and RNA their sequences. Now, since both proteins and nucleic acids contain nitrogen, let's compare amino acids and nucleotides side by side. How could you tell them apart? Well, there's a few different ways. Like that phosphate group is going to be a great clue because amino acids don't naturally contain phosphorus, and amino acids don't contain sugars. Ready to test yourself? I'm going to throw some tricky ones at you. Some of the comparisons that students find the hardest side by side. And if you're stumped, that's what practice is for. Don't worry, I'll come through with an explanation. Ready for the first one? Which one is a carbohydrate? Feel free to pause the video if you need a second to think about it. Otherwise, answer is coming in 3, 2, 1. So on the left, we have a linear monosaccharide, a carbohydrate. Do you know what's on the right? It's a saturated fatty acid, a precursor to many lipids. So how do you tell these apart? Well, first off, fatty acids will often be a good bit longer than the linear carbohydrates. And look at all those oxygens in the carbohydrate. We've got 5 carbons, 10 hydrogens, 5 oxygens. That's our 1:2:1 ratio. I'm not going to count up all of the elements in the fatty acid, but I can already tell there's not nearly enough oxygens there to give us that ratio. Next, which one is a nucleic acid? Again, pause if you need to. Answer in 3, 2, 1. On the left is RNA, a nucleic acid. Notice the repeating subunits, the nucleotides. You've got sugar, phosphate, nitrogenous base. Okay, but what's on the right? It's a polysaccharide, a carbohydrate. You've still got repeating subunits, but just repeating subunits of monomers, a much simpler structure here. Okay, you're doing great. One more. Which one is a lipid? 3, 2, 1. On the right side, we've got a steroid, a type of lipid. Notice the 4 fused rings. But we've also got fused rings on the left, so what is that? Those rings make up the R group of an amino acid, the monomer of protein. If you're feeling a bit more confident in identifying macromolecules like all of these, then give me a thumbs up. Be sure to subscribe for more biology tips and tricks from me. Check out my website for tutoring resources, and I will see you next time. Bye.